Hello everybody, and welcome to today's Revive webinar by the Global Antimicrobial Research and Development Partnership, GARDP. My name is Astrid Pence-Moore, and I'm hosting today's webinar about developing antibiotics for children, medical need, and regulatory challenges. This webinar is part of GARDP's education and outreach program, Revive, and Revive was launched in 2018. It aims to connect and support the antimicrobial research and development community by facilitating learning, connecting people, and sharing knowledge. Since the start of the webinar series in June 2018, we have hosted more than 30 webinars, and you can still watch the recordings of all previous webinars on our website, revive.gardp.org/webinars. Also, today's session will be added there in the coming days. On our website, we also host antimicrobial viewpoint articles by leading experts in the field of AMR. And just last week, we have launched a new resource, which is the Antimicrobial Encyclopedia. This encyclopedia currently hosts about 70 terms, key terms in the field of antimicrobial R&D, where each term has, at the very least, a short definition and a few carefully selected links for further reading. Some terms also have short explanatory videos by our experts. It's important to note that this resource will grow over time, so we will regularly add new terms and also more video content. We also appreciate your feedback, so we would be very happy to hear back from you what you think about this resource, which terms are missing. You can share links with us which we should add to certain terms. And even if you see a term where you don't agree with the definition, you can feel free to reach out to us and we will discuss this. Coming back to the webinar now, as always, the presentations will be followed by a Q&A session. So you can submit your questions in the questions window in your webinar control panel at any time during the webinar. And after the presentations, we will do our best to respond to as many questions as possible. I would also ask you to please address your questions to a specific speaker if possible. And today's speakers are Phoebe Williams from the University of Sydney and Sydney Children's Hospital, and Irja Lutsar from the University of Tartu in Estonia. Our moderator today is Sally Ellis, who is the project leader of GARDP's antibiotics, children's antibiotics program. And with this, I'm welcoming the three panelists and especially welcome Sally. I'm handing now over to you to introduce our first speaker.
Thank you, Astrid, and welcome everybody to this webinar on developing antibiotics for children. I would just like to introduce our first speaker. It's Phoebe Williams, who's a consultant paediatrician who completed a university medical training at the University of Sydney and Sydney Children's Hospital, and as well as taking further training in infectious diseases after completing her undergraduate studies in development economics. Phoebe has a diploma in child health, a master's in global health science, and a PhD in paediatric clinical infectious diseases, which was completed at the University of Oxford's Tropical Medicines Network unit in Kilifi in Kenya. Phoebe has worked both as a paediatrician in urban and rural health settings throughout Australia, but also frequently working in East Africa, in Kenya and Uganda. And she continues to work closely with a number of hospitals in low-income countries and is passionate about the global health issues that affect children. She'll commence an NHMRC fellowship aimed at addressing antimicrobial resistance in neonates in lower middle income settings, with a particular focus on the Southeast Asia region. I'm now happy to welcome Phoebe. Phoebe, please start your presentation. Thank you.
Thank you so much, Sally, and thank you, Astrid, for having me today. I feel really honoured to be asked to contribute to what's been a really fantastic webinar series that GARDP have hosted this year. Good morning to everyone joining us from Europe and the Northern Hemisphere. I'm joining from Australia, so good evening to anyone who's joining from down under. Today we're going to be talking about antibiotics in children and the development pipeline. And before we do that, I thought it was important to first look at where the medical need is. I'm just trying to click through my first slide. Sorry, it's a little bit slower linking from Australia. But if we look around the world and look at why are children less than 5 years of age dying at the moment, this data that's really beautifully painted by the Lancet Global Burden of Disease study shows us that across the world, neonates are by far the most at-risk age group of dying. You can see that box there where they constitute certainly the riskiest period in a child's life. And then infectious agents are really responsible for a vast majority of deaths across the world globally still today. So lower respiratory tract infections, diarrheal illnesses, malaria, protein energy malnutrition, which almost always is linked to death from an infectious pathogen, are really the main causes of death across the globe that we're still seeing today. And you can also see, looking at the shading of these boxes here, that we've not made huge gains in addressing these causes of death since the launch of the Millennium Development Goals in the 1990s, which obviously have now become the Sustainable Development Goals. But you can see the lighter shade boxes are the ones that show we have made some progress, but in general, many of these causes of death in children and neonates across the globe are yet to be really well addressed. And if we zoom in a little bit more closely on the neonatal causes of death, sorry, jumping ahead too fast. If we look at this pie graph here, it sort of takes that box from neonates and looks at why that neonatal age range, so obviously children in the first 28 days of their life, why these children are dying. And obviously intrapartum related events, so things that happen at birth, are responsible for some of those causes of death. But we can see that really a number of those causes of death are still due to infectious agents in that neonatal period. So prematurity, which is very often stimulated by an infection with a bacterial pathogen, as well as congenital pneumonia, neonatal sepsis and meningitis and tetanus, are really responsible for a number of neonatal deaths across the globe. So the neonatal age range is the highest at-risk period for a child, and even if they survive that period, then the chance of dying from an infectious pathogen is still the most likely cause of death in the younger age group across the world. And what we also know is that on top of this infectious burden, we now have the added issue of antimicrobial resistance, which is causing further neonatal mortality across the globe. And this is particularly implicated in low and middle income country settings. So multi-drug resistant pathogens, so that's those that are harbouring extended spectrum beta-lactamases, or ESBLs, you often see them referred to, and carbapenemases account for 30% of all global neonatal sepsis mortality today. And we know that low and middle income countries in particular are impacted by the emergence of antimicrobial resistance. And that's because they have a higher burden of infectious diseases. They also have weaker health systems where overcrowding might be very common, and there's often insufficient access to effective antibiotics when first and second line therapies fail. So this is one of the really sentinel studies that first tried to quantify how many babies are actually dying every year due to antimicrobial resistance. And as we can see, in India alone, it's estimated that approximately 60,000 babies are dying every year due to infections with antibiotic-resistant bacteria. And the other really interesting thing that this publication revealed was that across these high-burden countries that are documented here, gram-negative bacteria, which are reflected in the red graphs here, are responsible for a large number of deaths, and in many settings are actually outweighing the gram-positive bacteria that can cause neonatal sepsis. So historically, we always used to think of neonatal sepsis as being caused predominantly by organisms such as group B streptococci, but we're now seeing gram-negative organisms like E. coli and Klebsiella species causing a huge burden in neonatal sepsis, and that's certainly got an increasing burden with the emergence of antimicrobial resistance, which are really implicated in these gram-negative organisms. This was another fantastic study that came out the same year that looked at 3 tertiary neonatal units in India that was published by the Dennis collaboration. And the important thing that this study showed us was that across tertiary neonatal units with a very large number of babies being born, we can see that the organisms that are causing sepsis in the first 8 days of life really aren't changing very much. So again, historically, we used to think of neonatal sepsis as being something that was categorized as either early onset sepsis occurring in the first 72 hours of life, and that was historically thought to be due to pathogens acquired in the genital tract during delivery, and late onset sepsis, which was thought to be due to infection from pathogens acquired horizontally or in the environment. But what this study found was that the pathogens that are causing sepsis in neonates in their first week of life are really quite similar across those first 8 days. And it really led us to start questioning whether these definitions of early onset sepsis and vertical acquisition versus horizontal acquisition are accurate. And perhaps it is time that we start to think about the other ways that babies might be getting infected with some of these organisms, such as very early in their life in delivery suites that might be overcrowded and might have poor infection control policies, and how we might be able to therefore intervene in that period to minimise the acquisition of multi-resistant gram-negative organisms in these babies. I'm sorry about the delay that's happening with my slides clicking forward from down here in Australia, but thank you for your patience. So now looking at another continent, this was some research that I was involved in sub-Saharan Africa that looked at the rates of resistance to first-line antibiotics as per the World Health Organization. And that showed that there are very high rates of resistance to ampicillin, gentamicin and ceftriaxone among many pathogens causing sepsis in neonates and children. So you can see there that looking at, by this systematic review, looking at Klebsiella species and E. coli, for example, there was very high rates of not just ampicillin resistance as would be expected for those organisms, but also high rates of resistance to aminoglycosides and ceftriaxone, with half of Klebsiella species being resistant to third-generation cephalosporins and 16% of E. coli species being resistant to ceftriaxone. So in many of these health settings where that's the highest level of antimicrobial available, that really has a significant impact on the survival of these infants being infected with these organisms. And then this year, this paper from the NEO-AMR network, which is a huge network across many high, middle and low income country settings, published further elucidation of the epidemiology of infectious agents causing sepsis in neonates. And again, concerning levels of resistance in gram-negative organisms to third-generation cephalosporins, as you can see in the first column there, as well as to carbapenems. And I've highlighted some of those countries there that have quite significantly high rates of gram-negative resistance in those organisms. And then even in some countries, this is further added by the burden of resistance in their gram-positive organisms. So you can see in Nigeria, for example, a very high rate of vancomycin-resistant MRSA was elucidated alongside quite significant gram-negative resistance in their infectious pathogens that are causing infections in neonates. So we're gradually seeing across the globe a really increasing development of antimicrobial resistance in many health settings. And then this paper also came out this year, and again, similar to the data that was published in 2016, tried to elucidate the link between antimicrobial resistance and neonatal mortality. And when we look at these gram-negative resistant organisms, at these gram-negative organisms and the case fatality rates that are implicated for many of these babies, we can see that across these gram-negative organisms, there were very high case fatality rates at these units in Vietnam. And this cohort study really led the authors to conclude that first-line antibiotics were often ineffective in patients with healthcare associated infections, which was the pathogen, which was the cohort that was followed in this study. And that's due to delayed appropriate treatment. And in many of these health settings, there's not just a delay in being able to ascertain appropriate treatment, but even when there is the realization that the infant may have an infection with a gram-negative resistant organism or a gram-positive resistant organism, the agents that they might need to use to treat those infections simply aren't available. So the authors concluded that the absence of effective drugs in cases resistant to all tested substances contributes to the increased mortality in our cohort, since empiric treatment might fail due to antibiotic resistance. But we know that this is not just an issue that's afflicting low and middle income countries. While there's a lot of data that is fortunately being focused on those areas because that is the area of greatest need, we're also seeing data coming out of high income settings. So this publication in The Lancet last year looked at data from the European AMR surveillance network and found that the burden of infections due to multi-drug resistant organisms has increased significantly in the past decade, with carbapenem resistance, as an example, increasing from 18% in 2007 to 28% in 2015. And the size of these bubbles over here indicate the burden of resistance due to a number of different resistant organisms, and we can see third generation cephalosporin resistant E. coli causing a significant burden of disease there with the largest bubble, as well as MRSA, and down here this box that's the inset zooms in on the carbapenem resistant organisms and really indicates that we're seeing such rapid acquisition of resistance across so many health settings across the globe, including these high income country settings. Sorry, my slides have just frozen. So the authors concluded that each year 700,000 infections with multi-resistant organisms are occurring in the European AMR surveillance network, which is equating to 33,000 deaths every year. And to just put that into perspective, the disability adjusted life years, so that's collating both the morbidity and mortality that these infections are causing, is equal to the combined burden of influenza, tuberculosis and HIV in the European setting. So a huge burden in both high and low income country settings that's occurring due to antimicrobial resistance. And once again, when that's stratified by age, we can see that yet again, the youngest children are the most vulnerable. So looking here at the age range where the disability adjusted life years add up across the population, we can see that far and away, children less than 1 year of age have the highest burden of disability adjusted life years caused by antimicrobial resistant infections. And once again, about 2/3 of them, even in a high income country setting, are due to gram-negative organisms, with the remainder due to MRSA and vancomycin resistant enterococci. So that just paints the picture for you to really elucidate the increasing burden of antimicrobial resistance that we're seeing on the backdrop of what was already really high rates of infectious illnesses in children and neonates across the globe. And now I wanted to turn to how this is further challenged by the context of a really limited development pipeline for new antibiotics in children. So if we look first at what antibiotics we do have available for treating children and babies, I wanted to bring your attention to the Dr. Ware classification, which was published in the 2017 Essential Medicines List for Children by the World Health Organization. And this categorizes antibiotics into 3 main groups. So we have access antibiotics, watch antibiotics, and reserve antibiotics. And as you can see here, we've got many of the narrower spectrum penicillins and some early generation cephalosporins in the access group. In the watch group, we have some anti-pseudomonal penicillins or the beta-lactam, beta-lactamase inhibitor combination antibiotics. And in the reserve group, we have some legacy agents that have retained some very high efficacy against many organisms, mainly due to their lack of use worldwide, which has meant that they've retained good susceptibility, and also some of the much later generation cephalosporins. And these groupings are really important because what we want to see is healthcare systems using predominantly access antibiotics, with their prescribing practices. So access antibiotics are ones that have a narrower spectrum and also have a really good safety profile with a low toxicity risk in children. And these should be antibiotics that are not just the first or second line therapy prescribed for most clinical indications, but they're also antibiotics we want to make sure children do have access to, Because one of the real knife-edged swords of antimicrobial resistance is that while we know that overprescribing is responsible for a huge amount of excess mortality due to the selection pressure that is driven by excess use of antibiotics, we also know that there are hundreds of thousands of children dying every year due to the lack of access to simple antibiotics like amoxicillin to treat lower respiratory tract infections. So access antibiotics are antibiotics that we do want to have accessible in health settings across the globe. And ideally, given their narrower spectrum and their low toxicity risk, we'd like to see these antibiotics accounted for as approximately 60% of total antibiotic consumption in healthcare settings. The next group of antibiotics are the watch group, and these are antibiotics that are second choice treatments for a specific and limited number of indications, and they tend to have a slightly higher toxicity profile, and we also know if they're overused, they will drive the resistance potential. And this group also correlates with the highest priority agents that are published in the critically important antimicrobials for human medicine list. So we know that these are very useful antibiotics, but also antibiotics that are at a relatively high risk of selection pressure if they're overused. And the final group of antibiotics are the reserve group antibiotics, and this includes antibiotics that are really tailored to highly specific patients and settings that should be last resort options for life-threatening infections due to multi-drug resistant organism infections. And while these antibiotics can be extremely important for treating these drug-resistant infections, we also know that they need to be watched very closely with stewardship programs to ensure that they are also not overused and resistance is driven by their excess use. So that 2017 EMLC publication was recently updated with the 2019 Essential Medicines List for Children, and there were some minor changes there with 3 new antibiotics that were added into the reserve group. So we have ceftazidime, avibactam, meropenem, vaborbactam, and plasmamycin that were added into the reserve group. And there were also some antimicrobials removed, and they're predominantly antibiotics that we know have a higher toxicity potential in children. The other thing that the 2019 EMLC publication included was classification of all antibiotics into the AWARE groups, so that we have now classification of not just the 37 essential antibiotics, but 180 total antibiotics that are now classified into access, watch, and reserve, which is really important for healthcare systems to be able to know how to use those antimicrobials and which they should be targeting their use towards. Because we know that the overuse of watch and reserve antibiotics will exacerbate antimicrobial resistance. And this publication looked at how different health settings across different countries are prescribing their antibiotics and what proportion were prescribing predominantly access antibiotics. So remember, we want to try and have that set at a target of achieving 60% of total use within a healthcare system. And you can see the countries on the left of that graph are really close to achieving, some of them even are achieving that 60% target. But we can see that there are many countries that are really over-prescribing the watch and reserve antibiotics. And if we don't control this quickly, we'll see antimicrobial resistance continue to become an increasing problem across the globe. And that's really important because we know that at the moment, antimicrobial resistance is accelerating at a speed that is much faster than our development pipeline is being able to address it. This publication by the Pew Charitable Trust that I'm sure many of you have seen this year looked at how the antibiotic pipeline has proceeded over the last half a decade and found that really so many antibiotics that start phase 1 and phase 2 clinical development don't make it through into the later phases of development, and certainly even less make it through to the licensing phases. And even those antimicrobials that are proceeding along the development pathway, that are managing to get through those phases of clinical development, aren't necessarily going to be able to treat our most multi-drug resistant pathogens. So we have a very thin pipeline for antimicrobial development, and this is even more of an issue in children. We know that in children, paediatric drug development is particularly slow, and on average, the licensing of medications for use in children is currently lagging the licensing in adults by a decade. And that's due to many reasons, and they include a poor financial incentive from the market. So it's a very fairly unprofitable area for pharmaceutical companies to invest in. So there are less trials conducted overall. There's also a greater liability risk investigating this population, which deters industry from sponsoring trials. And there's a high risk of experiencing adverse events for this population to be investigated. And that not just adds to the liability risk, but also adds to the cost and the resources that are required to run these clinical trials, because when you're investigating a very sick and vulnerable group of patients, it's a really resource-intensive task for those clinical trial managers, and results in a lot of paperwork and can really slow down a trial. And this is all occurring in the setting of a complex regulatory framework. So if we look here at the way that antibiotics are licensed across the globe, we currently have two frameworks that are in place in two different major continents. So in the US, the the Food and Drug Administration oversees the licensing of antimicrobials there, while in the European Union, it's the European Medicines Agency. And for licensing to occur in each of these settings, quite separate study plans need to be instigated by the pharmaceutical companies and the study sponsors. And so often you'll end up with two clinical trials occurring at once to be able to enable licensing across these two settings. And they might have quite divergent inclusion criteria or ethics requirements from the two continents. And that can really be a significant barrier to seeing the licensing of these medications in children, because of the resource and financial investment that is required to enable that to occur. And so there are really many steps in the paediatric drug development process that act as burdens to, to streamlining the process of paediatric drug development. And I've talked previously about some of the trial design and conduct challenges and the regulatory processes, but there's also ethical challenges. We know that this is an inherently vulnerable population requiring investigation that have a high risk of mortality and morbidity, and it's very difficult to obtain informed parental consent from caregivers that are looking after a very sick child and going through an incredibly stressful period of their time, to be able to approach those caregivers and to know that you're getting informed consent from them when they might feel that they're pressured either way to enroll or to not enroll in a trial. It can make recruitment for these trials very challenging for those that are running the trials. There's also technical challenges in this age group that are quite unique. So when we think about something like blood volume collection, for example, and the process of collecting blood from a 1,000-gram premature neonate, and how much more difficult that is from simply collecting it from a well-grown adult, that requires quite skilled clinical trial staff to be able to run the trials. And it also can impact the recruitment of patients, because when you think, for example, for a PK or PD trial in children, how many blood samples are required to be collected and how that can even impact the blood volume on these very small neonates. There's many, many facets that need to be considered. And these children tend to require much more monitoring that would be needed for adult trials, and that's due to their really divergent physiology. We know that children possess a unique and really rapidly evolving set of physiological characteristics. Even in the first few days of life, the changes in renal function and drug elimination that occur in children are quite rapid and really divergent from day to day. And this means that there's big impacts on the ADME, the absorption, distribution, metabolism and elimination characteristics that we investigate for PK and PD trials in children. And that is therefore requires quite intensive resources compared to the very predictable metabolism that we see in adults. And so in the context of clinical trial design being so challenging and getting through the regulatory framework being such a hurdle in children, we're really seeing very high rates of off-label prescribing in children. So we know that nearly half of all paediatric medicines in Europe are currently prescribed off-label. And that means that they're prescribed in a manner that's different to what's suggested in the product license or is authorized in the product license. So you might see people using different doses or a different frequency, twice daily, 3 times daily, 4 times daily to what's in the product license. People might be using the drug by a different route, so intramuscular versus intravenous or oral, or sometimes even rectally, and also using the agent in a different age group to what it's licensed for. And this is all because we know that it's just so difficult to get this safety and efficacy data in children, and it's particularly a problem to get this data for neonates. But there's really currently a global consensus that we need to conduct these stages of drug development trials in neonates, as many disease conditions that are really only increasing in the context of antimicrobial resistance are specific to this population. And we know that we really need to try and ascertain better PK, PD and safety profiles in children, and particularly in neonates. When we look at off-label drug use in America, we can see from this publication last year that of 17,000 outpatient children that were assessed for their prescriptions, about 90% of the medications were prescribed off-label. And when we look in the bottom graph there, sorry, the bottom table there of the different drug classes that pertained to, we can see that anti-infective agents were by far and away the most frequently off-label prescribed medication for children in America. So off-label prescribing is really a widespread practice. And when we look at the agents that we have available for treating multi-drug resistant infections in children, and this review nicely summarised it, that was published this year, we can see that the off-label prescribing is really so widespread across the globe. So when we look at some of our available agents and the dosing recommendations that this paper pulled together, you can see, if we look, for example, at fosfomycin, that the dosing regimes differ from anywhere from 200 to 400 milligrams per kilo per day, so up to double the dose, with the frequency also quite unclear whether it should be 6 hourly or 8 hourly or twice daily. And when we look at the next column across at the age groups that have regulatory approval for many of these agents, we can see that some are licensed by the FDA for use in America, some are licensed by the EMA for use in Europe, and different indications are licensed in different regulatory areas, and that can really create a huge amount of confusion for the clinicians that are responsible for treating these multi-drug infections that are really life-threatening for a number of children. So what are the legacy agents that we have available for treating gram-negative multi-drug resistant infections in children at the moment? Polymyxins are still quite widely used in many, especially low and middle income country settings, because there have been some reports of efficacy when used to treat multi-drug infections in children. But unfortunately, these are really limited by their high rates of adverse events, particularly nephrotoxicity and neurotoxicity. And again, there's very unclear dosing regimes out there. The divergent case reports that do report treatment success in the literature all cite different dosing regimes, which creates significant confusion for the prescribers. And unfortunately, we're also seeing resistance to polymyxins increasing. And that's largely driven by the overuse of this agent in veterinary and agricultural sciences, and the runoff that has, and the ingestion that we as humans have through that interaction. Fosfomycin is another very useful legacy agent, which is a phosphonic acid derivative, so therefore in a class of its own, and has retained quite good susceptibility across the globe for many, both gram-positive and gram-negative organisms. And there was a recent trial by GARDP that will soon be published looking at the pharmacokinetics of fosfomycin in neonates, and we know that this is an agent that is likely to be best used in combination to try and limit the possibility of resistance emerging. Then there are some classes of antimicrobials such as aminoglycosides and fluoroquinolones, which still remain quite useful. So we know in many health settings, particularly higher income health settings, we still do often see susceptibility to amikacin or gentamicin that provides those agents as treatment options for children, but that's not necessarily the case in many low income country settings. And aztreonam and tigecycline used to be quite useful legacy agents that were used as a last resort for some multi-drug resistant infections, but these have been removed from the essential medicines list that was published in 2019 due to some concerns about their toxicity profile and their somewhat limited efficacy on balance with the toxicity that they can cause. And then we also have some newer agents for treating gram-negative multi-drug resistant infections in children. And these include carbapenems, which can be particularly useful for treating ESBL-producing invasive infections, and even some carbapenem-resistant Enterobacteriaceae infections. And that's because we know from Monte Carlo simulations that when these agents are used at a high dose and as extended infusions, they can often overcome some of that carbapenem resistance and still be a useful backbone agent. And then there have been a number of beta-lactam, beta-lactamase inhibitor combinations that have emerged over the recent decade. There's ceftazidime avibactam, which is now approved by the FDA for use in children more than 3 months of age with complicated intra-abdominal infections. Meropenem avibactam, which currently has a phase 1 trial underway in children. There's imipenem cilastatin relebactam, which also has clinical trials underway for its use in children, and is looking to be a good potential treatment option for multi-drug resistant Pseudomonas aeruginosa infections. Ceftolozane tazobactam, which once again will likely be something that is going to be helpful for multi-drug resistant Pseudomonas infections, and we currently have a PK and safety analysis underway in children. And finally, cefiderocol, which is a novel siderophore cephalosporin agent, which is also currently under investigation in children and has managed to reach a phase 2 trial for children as young as 3 months of age. So that provides a brief overview for you of really the setting of where the need for addressing antimicrobial resistance in children and the high level of infectious disease morbidity and mortality is occurring across the globe. And we know that to try and address this, we're going to need a really interconnected approach across academia and pharmacy and many of the global non-governmental organisations and multilateral organisations to try and address this. We know that AMR is particularly impacting children, and neonates by far and away are the age group that are most highly impacted by the emergence of antimicrobial resistance. And we know that we need access to effective access antibiotics and usage in all health settings concordant with the access, reserve and watch categorisation is vital for us to stop further antimicrobial resistance emerging in children. So it's important that where we can have them, antibiotic stewardship programs are put in place to try and protect the watch and reserve categories of antimicrobials that we have. And to ensure efficacious treatment options remain available for children, we need to try and see the licensing process streamlined. And one of the main aims of the coming decade should be to try and minimise the big time lag that we're seeing between the licensing of agents for use in adults and trying to get them into the paediatric population as soon as we can, because this is by far and away the population that is most impacted by infections and certainly impacted by antimicrobial resistance. Thank you very much.
Thank you very much, Phoebe, for that very extensive presentation and sort of covering some of the issues around antibiotic use in children, but also some of the challenges with addressing that. I'd now like to introduce Professor Irja Lutsar. Irja is a professor in clinical microbiology and head of department at the University of Tartu, Estonia. She gained her medical degree and PhD at the University of Tartu and is a qualified paediatric infectious diseases specialist. Between 95 and 98, she was a research fellow in paediatric infectious diseases at Southwestern Medical School in Dallas, USA, and also has spent a significant amount of time working as Director of Clinical Development at Pfizer in the UK. She's got extensive research interests, including in antibiotic pharmacokinetics and dynamics, neonatal infections and fungal infections, and antibiotic resistance. She's been leading investigator in more than 20 clinical trials in children, including studies evaluating the pharmacokinetic properties of antibiotics in premature neonates, and has published more than 150 publications in peer-reviewed journals. And she's also a member of the Paediatric Committee of the European Medicines Agency since 2007. Welcome, Irja, and please, can you now start your presentation? Thank you.
Thank you very much, Sally, for a nice introduction, and thank you very much giving me an opportunity to talk about clinical development of antibiotics in pediatrics. And I believe that the introduction for this talk was already made by Phoebe. She already mentioned or highlighted the problems we have in clinical development, and most importantly, the lag between the adult approval and pediatric approval, which is approximately 10 years. And oddly enough, the population that needs new antibiotics the most, these are the newborns or the babies or children below 1 year or below 5 years. They will be the least ones who are getting pediatric data. And I'm going to discuss a little bit what to consider in terms of pediatric antibiotic development, and most importantly, how to speed it up. So if you start, and I'm sort of talking of developing new antibiotics, but this very much applies also to repurposing antibiotics. So if you start pediatric development, the very first thing you need to think is you need to think, is this new agent or new antibiotic needed in pediatrics? Or what is the indication? Is the indication the same in adults and also in pediatrics? Or whether this new treatment is so-called me too, or there is no unmet medical need. For example, for otitis media, there are sort of plenty antibiotics already in the market, and some of them quite effective. So if your antibiotic does not add anything, then maybe that does not need to be developed for pediatrics either. And is this antibiotic needed in all pediatric age groups or only in some pediatric age groups? And again, if this drug is sort of good inhaled antibiotics, most likely you would not need, at least that's current thinking, that this being developed for neonates, but it's again very much needed for patients with cystic fibrosis, for example. So if you have got answers to for first question, you go to the next steps, and you think what is known about this new antibiotic in adults. Has the PK/PD target been characterized and defined? And do we know the efficacy and safety in adults? So here is a graph describing how you consider or how you walk through the pediatric drug development program. And there are two options. The first one is you do the full development program, and we go back to that later in this talk. Then that means that you need to do PK studies, PD studies, you need to do efficacy and safety studies. Or you can also choose to do limited program. When children are similar to adults, and then you need to do only PK/PD study, or in other words, to define the dosing recommendation or dosing regimen, and you can extrapolate. Everything else can be extrapolated from adults. So if you have to do the full program, the full program usually is required when these antibiotic or anti-infective agents is indicated for predominantly childhood infections, or where the infection does not occur in adults or cannot occur in adults, like congenital CMV infection, obviously not an adult disease. So in those cases, you need pediatric development, also for otitis media, which is mostly pediatric disease and very rarely occurs, or at least does not occur in similar way in adults. So if you go to full development program, then this program obviously needs to be conducted in pediatric population or in target population. First, you start with the PK studies because you need to define the dose first, and then you most likely will be requested to do efficacy safety studies that have adequate power for efficacy. before embarking to pediatric studies, you still need to make sure that you know enough of these antibiotics from adult trials. So whether antibiotic, and most importantly, we are most importantly here concerned on safety. If there are already safety problems in adults, many antibiotics don't even reach market when they have safety problems. But if there are specific safety problems, then this is something that very carefully needs to be monitored in pediatric trials as well. And more, the pediatric committee of the European Medicine Agency encourage companies to do parallel development. So if you already have first data in pediatric phase 1 trials, you already start thinking how I will develop this drug in children. And the reason being that if we do the staggered approach, it may not be justified, because very often you can't actually extrapolate data from adults to children, and then the pediatric development is significantly delayed. So currently it takes about 10 years before the drug dosing recommendations or the indication will be approved in children, and this is certainly not acceptable in case of antimicrobial resistance increasing. So the full development program, I'll just bring you some examples. Community-acquired pneumonia may be very different in children and in adults. You have seen that many cases of community-acquired pneumonia in children are caused by viruses, or ventilator-associated pneumonia is also something which is very different in children and in adults, and it's going to be increasingly difficult to do studies in ventilator-associated pneumonia, because this is fortunately becoming very rare disease in pediatrics. Acute otitis media, as Rodi mentioned, is not adult disease, it's a sort of pediatric disease. Or group A beta hemolytic streptococcal tonsillitis, which is yes, self-limiting disease sometimes, or does not require antibiotics, but studies have very clearly shown that the effect of antibiotics is worse in children than it is in adults. And it's probably not that difficult disease to study, because it's quite common. And the neonatal, there are very few antibiotics where you have indication in adults as a sepsis, whereas in neonates, most cases, we come back to that, are actually neonatal sepsis. And the fact that the neonates are immunotolerant and may require different antibiotic exposure than adults is also very well acknowledged. So, in order to speed up the pediatric development, the concept of extrapolation is more and more introduced, or more and more used in pediatric drug development. And for extrapolating, if you look at the graph to the left, you can see that then more points you have, or in other words, then more knowledge we know on physiology, on pharmacokinetics, and pharmacodynamics of antibiotics, then better are we in extrapolating. So there is an arc. If you only know 2 points, then obviously you can draw the straight line here. And but if you have more points, you actually would see that this is arc. And this is actually where the problem lies. We just do not have enough knowledge on PK, PD, on physiology. We need more basic knowledge in pediatrics and also in adults before we can go 100% to extrapolating. Yes, I agree that maybe sometimes in the future, not in my lifetime, but later on, we can do drug development in silico, or in sitting in the front of my computer, as I'm doing right now, but we are not just there. Our basic knowledge is not that good. So if we go to there is also guidance documents on extrapolation, and regulatory agencies are actually very much supporting extrapolation. So there is a big change has happened within last 10-15 years in regulatory field as well. So we acknowledge that this enables us to avoid unnecessary studies in pediatrics. This allows us to allocate resources to areas where studies are the most needed, and this also allows us to conduct studies in situations where the feasibility of studies is restricted, as already mentioned by Phoebe in her talk as well. So how do we can extrapolate? There are sort of two main ways in this extrapolation approach. So one is the extrapolation, extrapolating efficacy from studies in adults, provided and looking that we are targeting pediatric exposure similar to drug exposure in adults. So basically, if the disease in children and adults is similar, then targeting or getting the same AUC in most cases, or getting the same drug exposure in pediatrics, so we can avoid doing, or we can prevent doing efficacy trials in children. We just extrapolate the efficacy, and in some cases we even can extrapolate safety, as I come back to that as well. The other way of extrapolating is this PK/PD based extrapolation. So if we know clearly the PK/PD of the new antibiotic or old antibiotic in adults, we can do the target getting the same probability of target attainment in children, and then that's another way extrapolating from adults. So if we want to extrapolate efficacy from adult studies, then we assume that the infecting organisms in adults and children are the same, and very often microorganisms don't know whose organism they are causing infection. And we also assuming that disease process in adults and children is the same. This is often actually the case in the field of infectious diseases, not necessarily in other fields. So in that case, we need to do the PK studies in children, because PK is always going to be very different, and especially we need to do studies in neonates or smaller children, and maybe not necessarily in adolescents, but it sort of is agent dependent. And then if we do the PK studies using AUC or the AUC approach, we can actually target the same exposure in children and extrapolate efficacy. And depending now on the regulatory agency, the safety can be also in larger part extrapolated, but the smaller safety trials may still be needed in pediatric population as well. The problem here is that if the pediatric and adult indication for antimicrobial is different, it's difficult to extrapolate. And what we are seeing, I brought already example like pediatric CMV disease or congenital CMV disease. You really can't extrapolate the CMV in adults, which mostly occurs in immunocompromised adults, to children with congenital CMV. So if the disease process is not the same, you can't really do extrapolation from adults. And then the other way is to do the PK/PD based extrapolation, or you are targeting the PTA in pediatric patients. So you assume that infecting organisms in children are different or have different resistance. So you can't really directly extrapolate from adult efficacy trials. So you also assume that the minimum inhibitory concentration in pediatric microorganisms are known, and this is not very often the case. That most MIC studies are done actually with organisms isolated from adult population. You assume that the disease process is very different, and as I already mentioned, that in neonates, the neonates who are immunotolerant, they may require higher antibiotic concentration because their immune system doesn't work as well that it does in adults. And you also assume that you know the PD targets, which is either AUC above MIC or time above MIC or Cmax above MIC. So because of those all PD targets you all know very well. And then you do the PK studies, you define the dose, and you show how the drug behaves in that target population or pediatric population. And then you, by using multi-canal simulation, you most likely take that's where the problem is, that you select the MIC that is the highest MIC value that this patient population or this microbial population has, or basically you take the MIC breakpoint and you take also the maximal PID index. And then the problem here is that do we really need to take the highest MIC value? Because the highest dosing or highest AUC or highest dose may also cause more safety issues. So and nobody really knows exactly whether we need to take the highest MIC value or we need to take the sort of mean MIC value, and whether we need to use the maximal PD index, for example, for beta-lactams, whether we need to assume that the time above MIC should be 100% always, considering that neonates are immunocompromised, or we can accept lower values, 50 or 60%, that we do in adults. So I briefly already touched that one, so we can skip this slide in interest of time. And I just bring you an example in piperacillin, tazobactam in oncology patients with fever and neutropenia. These patients, of course, are immunocompromised. And if you look at this graph, if the agent is very susceptible, has MIC values 0.25 or 0.05, and this is actually the probability of target attainment. It is agreed that you want to have this 90%, although some regulatory agencies could accept 80%. But let's say we want to get best for our children, we have this probability of target attainment 90%. And if you have very susceptible microorganisms, MIC up to 1, it really does not matter what dosing regimen. And here are different dosing regimens listed. It really does not matter what you use. This antibiotic is going to work anyway. But in this case, we are talking of more resistant organisms in conditions where we have limited option for treatment. So sometimes we need to use these beta-lactam antibiotics, and by increasing their dose or by improving their dosing regimen, then we can use even old antibiotics, even in conditions where the MICs are relatively high. And here you can see the example of with the black dots. This is an example of continuous infusion, 40 mg per kilo, relatively high dose of continuous infusion. You can see that 90% of probability of target attainment can be achieved for quite a high MIC. So this is something you can actually do in silico, and to at least to have some dosing regimen for testing. I'm not saying that we are quite ready there, that we can just work out the dosing in computer or in silico, but this is just something that then can further be tested in clinics. So then there is another area which is increasingly coming or increasingly being more important is pediatric development in cases of limited development in adults. So although the multi-resistance is increasing, there are some multi-resistant organisms that fortunately are extremely rare. And even in adults, you can't do fully powered efficacy trials because you don't just have enough patients, or you may have enough patients worldwide, but to do trials with 100 or 500 centers worldwide, it's logistically extremely difficult. Or if some very rare infections in heavily immunocompromised patients, as we see in our clinics every now and then. Then if the development in adults is already limited, or fully powered efficacy trials are not requested in adults, then of course they are not requested in children either. So in those cases, you only need to do PK data, and very limited safety is required in children, and mostly the efficacy, or mostly the how the drug is going to behave, is extrapolated from adults. So now, establishing the safety, and it has been sort of golden rule that safety trials are always required in children. And this is very true for the American development, or for the FDA as well. But the European Medicine Agency is more and more sort of questioning whether the safety trials are always required. And the question is, the reason there being that the underpowered safety trials show very little, and I really recommend you to read the paper that is referenced here, that actually shows that the overall safety of antibiotics is predictable, especially in those classes that have been long time in the market. The new cephalosporins that are coming into the market haven't shown anything that we haven't known already before, and whether we can also extrapolate safety from trials conducted in adults from our past experience. This is currently under discussion. So ideal trials for the safety would, of course, be the comparative studies where you have comparator group and also the study group, and if they are sufficiently powered. This, however, could delay pediatric drug development significantly. And often these safety trials, even if they are requested by regulatory agencies, they are very small. They usually have 50 to 70 patients. That does not really show you the safety concern if they are not extremely common. And more nowadays, we need to explore possibilities of using registries or clinical databases in order to observe safety after the drug has already in the market, or after the drug is already sort of given limited indication in pediatrics. I think that this is unused resources at the moment, because every hospital has nowadays these electronic health records, and this material is completely unused in terms of drug development. So my very last slides are a little bit on drug development in neonates. As you all know, in PK in neonates is very different of that in adults, and you really can't extrapolate the PK data from adults to neonates, and the reasons were already mentioned by Phoebe in her presentation. The other thing is what is different in neonates is the neonates have this so-called unspecific infection, most neonates have the neonatal sepsis, either late onset or early onset sepsis, and I very much agree with Phoebe that probably this is outdated definition, this early onset and late onset. We need much better definition, and this does not make sense anymore to divide the early onset and late onset sepsis, pediatric neonatal sepsis. And therefore, we need the drug development or the PK studies. Of course, you can't do PK studies with antibiotics in completely healthy neonates. The PK studies also will be conducted in pediatric patients with neonatal sepsis, or at least with suspected neonatal sepsis, who would require antibiotic treatment. So I'm not sort of touching the space here too much, showing that more than half of the cases of neonatal sepsis have been, at least in developed world, and been caused by gram-positives, but quite significant number of gram-negatives is causing neonatal sepsis as well. So for the neonates, we need to bear in mind that we need to enroll term neonates and also preterm neonates, because again, PK of a preterm neonates, very preterm neonates, is quite different of that in term neonates because of the immaturity, most importantly. Then the condition that you would study in neonates would be the late onset sepsis, or neonatal sepsis is probably the right definition. You would do the PK studies, and regulators encourage to do these so-called add-on therapy studies, which are also not the easiest studies to do, but you don't want to give the new drug as a single agent to a neonate when you don't exactly know how this drug would behave. So add-on therapy is preferred. You can't take too many samples from a neonate, as already mentioned by Phoebe as well. So the sparse or semi-rich sampling is encouraged. Microsampling assays are developed, and you also want to accommodate your clinical research with the everyday clinical practice. So if the sample is taken for another for clinical indication, then laboratory leftovers could be used for the PK studies. It's logistically more difficult, but it's sort of more acceptable by parents and by families as well. And the staggered approach is not necessarily always, or you always need to think, if you go to the staggered approach, there is a lot of emotions in going to the staggered approach, but there is a very little scientific justification to go with the staggered approach. It's the problem with the staggered approach is that it significantly delays neonatal drug development. And this is my final slide. There are very many stakeholders in the pediatric clinical development. There is certainly academia and non-profit organizations like GARDP. Their role is to actively participate in the studies, to design the studies, but also to identify pediatric needs. And then also it's up to the pediatrician to say that, no, we don't want to use not studied or off-label, we don't want off-label use in pediatrics. This is not acceptable in 21st century. There is also the regulators play a significant role, demanding the studies in pediatrics and being actively participating in approvals of antibiotics in pediatrics, and a huge role is played by the industry to develop drugs or new agents in pediatrics, despite that there are not that many incentives, and that may not be profitable to develop child-friendly formulation and most importantly, timely conduction of pediatric studies. And I thank you all very much for listening.
Thank you very much, Irja. We will now start the question and answer session. So just as a quick reminder for the audience, you can submit your questions as shown on this slide in the on this slide in the question box. Please include the name of the speaker you're addressing your question to. We will do our best to respond to as many of the questions as possible during the allocated time. Just for the initial questions, maybe we'll start with Phoebe, but then maybe also Irja can comment on this as well. With respect to the antibiotic development pipeline, are there common reasons why many antibiotics fail to reach the market? This is more general. Yeah.
There's really many steps in both adults and children that antibiotic development falls off the development pipeline. Certainly this year with COVID on board, we've seen trial recruitment be a huge problem. Many people have been unable to reach their recruitment targets for clinical trials, and that's resulted in some trials having to be terminated early. And that is a problem across both adults and children, but also particularly in children, because often the trials are really targeted to such specific clinical indications that the recruitment target is quite hard to achieve for the number of children that might have a specific clinical indication for that clinical trial that is recruiting. So being able to move through the phases of development requires a huge investment from the sponsors and from the industry that's running the trial. And as soon as there are challenges with reaching recruitment targets and with any safety signals that might come out, that further can exacerbate those challenges. And I think that that's something that's common to both adult and paediatric trials, but is a particular problem for paediatric clinical trials, in that so many of these agents are recruiting for such narrow inclusion criteria that really restricts, when you add in an added challenge of something like coronavirus, it can really restrict being able to achieve the recruitment that's required to generate the safety and pharmacokinetic data that's necessary.
Yeah, if I may add to Phoebe also that what I have seen over the years, that the people are increasingly more busy. So in many hospitals worldwide, people are very busy in their clinical work, and we really don't have, or the sort of medical community does not have enough, does not give enough time for people to do research, and this is certainly one reason, and it's even more difficult right now, where everybody is busy doing COVID. And the other thing is that we need more training in pediatric clinical research. So people are very optimistic going to clinical trials, that I do the PK trials, or I do the neonatal sepsis trials, because I have plenty of patients in my hospital. But when the trial starts, then people realize that the clinical research and everyday practice is not exactly the same. Those patients whom I give antibiotics everyday practice, they do not qualify to clinical trial. And I think that we need to start or improve our training from the university onward, and then postgraduate training as well. So we pay very little attention in the universities in training our students for clinical research, and I see that that's where it all starts. And then of course it's the main reason is that the trials, they are planned to do within 18 months, but it actually happens to be more like 5, 6, or 7 years, and then they could only recruit half of the patients they planned. So those are the main reasons I see.
Yeah, no, thank you both of you. I mean, I think one of the other challenges is particularly there is not really a market well, there's definitely not a market for antibiotic use in children. but also there's a problem with adults as well, and particularly a lot of the antibiotics that are being developed these days tend to be by smaller biotech companies, and they just don't have the capital to move particularly into paediatric development. So one of the challenges that GARDP particularly is trying to address is to try and facilitate that process. But as you've heard both from Irja and Phoebe, it continues to be a challenge to move sort of newer antibiotics forwards into the development. So maybe leading on to sort of the next question, which I have to say is always a challenge, whatever you're doing when it comes to clinical trial. Is it possible to perform paediatric studies with enough power to show statistical significance? I will probably focus more on the PK, because I mean the efficacy and safety is already going to be a sort of a significant challenge anyway in terms of the name. In terms of the numbers that are required, and then what can be done to improve this power?
Yes, and in terms of PK trials, I think that it's possible, but you need to really have people who have prior experience of doing PK/PD trials. If in your hospital nobody has done PK/PD trials before, or is even not, they are very challenging trials, and you need the sort of you need whole staff. You need research nurses, you need lab technicians, you need also interested doctors. So I think that in those cases it's possible, and most importantly, you need time. You can't start doing clinical research at 5 o'clock or 6 o'clock in the evening if you have had busy clinic and several ward rounds and many complicated patients in your ICU. You really need dedicated staff. In those cases, it's possible. It is possible to combine also clinical, or it should be possible to combine clinical work with the research, but it's not that the research comes on the top of your busy clinical work. So that's, I see, major problem, that people are sort of overwhelmed with all sorts of responsibilities.
Yeah, no, absolutely. Maybe Phoebe?
The only thing that I would add to that is that one of the sort of blessings that's emerged in pharmacokinetics in the last decade is a focus on population pharmacokinetic modeling, which has really enabled a shift from the sort of old style pharmacokinetic trials that required lots of blood sampling in kids, that were really quite technically challenging and ethically challenging to get past ethical review boards, and obviously also required a significant blood volume. And now with the use of sort of non-mem and other statistical packages that have enabled pharmacometricians to be able to contribute to, you know, the development of this pharmacokinetic data, I think that's one of the saving graces of clinical research in neonates in particular. But again, that's a skill that's really far too you know, underutilized, or there's just there's not enough people globally that have those skills to be able to link into these trials. But it's certainly one of the promising facets of being able to do PK/PD trials in infants if the funding is there.
Okay, so one of the other questions that I've got from one of the attendees is how has novel formulations of antibiotics like nano delivery systems impacted the development of resistance to antibiotic microorganisms? So I know maybe focusing more on sort of the more traditional formulation development, because I think paediatrics is always the last bastion of sort of the new technologies, and if it's not available in adults, then it's certainly not going to be available in children.
Yes, you're absolutely right, Sally, that if you talk to the industry or researchers or formulation researchers in the industry, they have fascinating formulations for children, but those are not very cheap. They are difficult to develop, they become more expensive, and most importantly, when they come into the market, the hospitals are not going to buy them. So that has been shown with the inhaled antibiotics. There have been very good formulation of inhaled antibiotics, mainly for patients with cystic fibrosis in the market. But hospitals just don't buy. They just say, okay, we have plenty of gentamicin, we just reconstitute that in some liquid and we get that very cheap. So we don't have money for this expensive formulation. So I think that there is a sort of you need to change your mindset as well. So in not only in pediatrics but also in hospital management and also on the level of taxpayers, that you know, medicine is not that cheap, and I think that the industry is able to do very nice formulations, but you need to consider that these formulations are not going to be the cheapest one in the market, and that's where I see the problem is. We still have these crash tablets.
Yeah, absolutely. And when you think about where, you know, the real burden of infectious morbidity and mortality is across the globe, it's certainly outstripped in low and middle income countries. You know, while there are issues with antimicrobial resistance in high income countries, the greatest absolute numbers of children that need treatments for these antimicrobial resistant infections are in healthcare settings that just can't afford these technologies. You know, these are settings that don't even have meropenem to grab in their cupboard for an ESBL or carbapenem resistant infection. So those sorts of technologies, while promising, they're probably not something that will make a difference on a population scale.
Yeah, and I think one of the other things, so one of the more basic technologies which is available, like oral dispersible tablets, they're available, particularly more, I'm certainly aware of it in low middle income country settings, but even then they're not necessarily used as widely as they should be. The focus is still very much on the sort of traditional either tablets or syrups, which is already acknowledged that, I mean, WHO recommends the use of oral dispersible tablets as routine, but when you actually look at what is actually being used, in most cases, it's still syrups.
Yeah, absolutely right. The sort of the approach we always have done like that. We always crush the tablets. So
yeah.
don't look that there are actually better formulations.
Okay, maybe a couple of one or two more questions. So we've focused quite a bit on sort of antibiotics, but there are also other sort of key ways that we could think about that could improve the current burden of neonatal sepsis globally. That are relatively basic as well. Maybe you'd just like to comment on that. Maybe to Phoebe first.
Yeah, absolutely. You know, while we're never going to really catch up with the rate of AMR emerging with what's in the antibiotic pipeline, I think it's really important that we look at really simple infection prevention and control mechanisms that can limit the spread of antimicrobial resistance and the likelihood of particularly babies acquiring these organisms. You know, the Dennis collaboration showed us quite clearly that a lot of these infants are probably getting colonized with multi-resistant gram-negative organisms very quickly after birth, and whether that's because their mothers are colonized in their vaginal tract or whether it's because of inadequate opportunities for the staff to have hand hygiene between patients, or whether it's because they're in overcrowded facilities with you know, many babies side by side and no infection control, or ability to sort of separate those very overcrowded facilities, the babies and the mothers. I think there's a lot of really simple things that we could improve on, such as hand hygiene and focusing on enhancing breastfeeding and cord care, and, you know, kangaroo care. There's a lot of really simple and quite cheap interventions that could be really efficacious in a lot of these settings in diminishing the rates of antimicrobial resistance spread and acquisition for these babies, that could make the difference between you know, needing to even go down the treatment pathway.
Okay. Thank you, Phoebe. I don't know if Irja, if there's anything that you wanted to add.
Yeah, I just want to stress that the hospital hygiene and following this hospital hygiene rules and cohorting, testing, cohorting or sort of isolating those patients infected with antimicrobial resistant organisms might be sometimes even more important than the availability of brand new antibiotics. So that's extremely important.
Okay, so I think we've now really sort of come to the end of the question and answer session. I would like to thank everybody for the questions and comments, and once more thank you both to Phoebe and Irja for your excellent presentations and contribution to this webinar. I'll now hand back over to Astrid.
All right. Thanks a lot, Sally, for moderating the webinar, and thank you very much to Irja and Phoebe for your presentations. I also want to point out that we had quite the spread of time zones on our panel today. So for Sally in the UK, it was pretty early when we started, even before going live. And for Phoebe, it's now already her evening. So I think, I'm sure you're very keen to sign off and go back to your life, actually. I just want to take 1 or 2 more minutes to announce our upcoming webinars, where one specifically might be of interest for today's audience. So on the 17th of November, we will have a webinar about discovery of new antibacterials using artificial intelligence. And on the 20th of November, we have a webinar which we are developing in collaboration with the WHO about saving children's lives treating neonatal sepsis. So you can already register for both webinars on our website, as shown here, revive.gardp.org/webinars. And also on this website, you can always find the announcement of future webinars. So this is at the moment, this is all which is announced, but you can be sure that in January and the rest of 2021, there's a lot more coming. And yeah, that's all from my side. So again, thank you to our speakers, and thank you to our moderator, and also thank you to our audience for contributing with your questions. I hope this was interesting and useful for everybody, and that you join for our future webinars. Thank you, and goodbye, everybody.
Thank you.